Hi, I'm Schoenauer Sebag, and as just said, I'm very happy to be here today to present you with our generic methodological framework for studying single-cell motility in high-throughput time-lapse data. So this work was done during my PhD under the supervision of Thomas Walter. So today I'll briefly start by introducing why one could be interested in studying single-cell motility, before detailing you the workflow and presenting you with some results. So cell motility encompasses any cell movement which is active, that is energy consuming, and so it includes cell migration, but it's broader than that. It's a property that is involved in the epithelial mesenchymal transition, and as such plays a major role both in embryonic development and in the process of cancer metastasis. So for this reason, it's very important to be able both to know and to learn its molecular basis, as well as to detect environmental contaminants and drugs that can perturb it. So the usual approaches to study cell motility use wound healing assay or migration chambers, and hence provide the user with aggregated data at the level of the cell population. However, as is the case for most biological processes, cell motility can be very, very heterogeneous in a population, and that's indeed what you can see here on this video of HeLa cells, which have fluorescently tagged histone protein, hence that we can follow, that we can see, whose nuclei we can see. So in order to be able to differentiate those different behaviors, we really wanted to study cell motility at single cell level. And so for this, we use this very specific type of time-lapse data, which is video microscopy data. So following an initial perturbation, so cells are exposed to a drug or environmental contaminants or small interfering RNA, images are acquired over multiple time points, and the fluorescent tagging of the protein of interest enables us to follow what happens to them. So in this data set, so the MitoCheck data set, as in the previous slide, one histone protein enables us to see the nuclei, of the cells. And and in this data set, it was RNA interference screen, meaning that the cells have been exposed to a small interfering RNA, which downregulates one gene of their genome. So to be able to study cell motility in this type of data, we have developed Motif for motility study integrated workflow, whose goal is to study cell motility under chemical perturbation at single cell level and on a large scale, which is what high throughput means. So let's briefly see what it's about. So first of all, so the cells are exposed to some perturbation, as I was saying, either a small interfering RNA, a drug, or an environmental contaminants. The data is acquired. Following this, the objects on each image are segmented, so we identify the nuclei. They're quantitatively characterized. For example, we compute their mean intensity or their perimeter. This information will enable us to track the nuclei in 2 dimensions over time, which will produce a set of cell trajectories. Themselves will be described quantitatively. So for example, we're going to compute the mean square displacement of the cell on its track. And finally, we will use all those feature distributions to assess if single cell motility was significantly different in the experiment from the control videos. And this will give us a hit list of experiments in which we think that this process is different. And we can then go back and get a hit list of either genes whose downregulation perturb single cell motility, or of drugs or environmental contaminants that perturb this process. So let's see the workflow more in depth. So the 2 first steps, so namely cell segmentation and cell feature extraction, are done using Cell Cognition, which is an open source software that we co-developed with the IMBA in Vienna and the EMBL in Heidelberg. Once we have quantitatively characterized each nucleus in each frame, the question arises to know how one can use this information to be able to track the cells over time. And so given that we are working with cells that are under some perturbation, whose impact on cell motility we don't know, we really wanted to use the minimum prior knowledge to do cell tracking. And that's why we chose an approach of learning cell tracking from an annotated set, so manually annotated set of trajectories. So for this, we inspired ourselves from a paper by Lu and Hamprecht. And this enables us to learn cell tracking rather than a priori model cell motility or have to manually tune parameters. So very briefly, how does this work? Well, so we have already identified all the objects in the consecutive frames. And what we're going to do is that we're going to enumerate all matching possibilities between the two consecutive frames within a reasonable distance and under the constraint that no one's left behind. And given this annotated set of trajectories, well, we're going to globally match the two frames, choose the matching that's the most probable. So following this, we are here, and so we have for each experiment a set of trajectory features. We are going to be willing to describe them too, and so for this we have found in the literature and partly designed ourselves a set of 15 trajectory features. So first of all, we're going to be interested in particle motion information about the cell. So namely, for example, its diffusion coefficient. Other global trajectory features would be the convex hull that the cell covers on its track. And finally, other information will be average local features such as the mean square displacement. Let's see the example of the movement type. So under the very simple model that the distance a cell travels during time t is proportionate to t at the power nu, well, what we call movement type and what we're going to compute following the formulations in those papers is this power nu. So if nu is equals to 1/2, well, it's a purely diffusive motion, whereas if nu is equals to 1, it's a perfectly directed motion. And so one good thing when working with videos or images is that you can go back to the data and actually look at what you wanted to compute corresponds to something. So here you have HeLa cells coming also from the methodological data set, and each dot represents a trajectory that we've computed and that passed the trajectory quality control. And the color of the dot indicates its movement type, so the yellower the closer to 0, so the more subdiffusive, and the redder the closer to 1, so meaning the more directed. So if you look at the cells now, you can see that indeed, so cells that have a red dot are clearly more directed than cells that have a yellow dot. So if you look at this group of cells here, it's kind of obvious want to exit here. I mean, it's like, so it corresponds to what we wanted to compute. Once we have these 15 distributions of trajectory feature per experiment, we will want to use this information to differentiate, to identify and differentiate experiments from controls and to know if cell motility was different in the experiment. So what we're going to do is that for each feature, we're going to do a two-sample Kolmogorov-Smirnov test between the experiment and the control of the same batch. So this will give us 15 p-values, which we'll combine using the Fisher formula. There is no theoretical distribution of that the statistic follows under the null hypothesis, given that the tests are not independent. So how do we assess when the statistic is significantly high? Well, what we're going to do is that we're going to repeat the exact same procedure, but we're going to compare control to control experiments and hence get a distribution of the statistic under the empirical null hypothesis. That is, we really don't want that one control is significantly different from another control. So this gets us a distribution, empirical null distribution of the statistic, and we can then compare the statistic obtained from comparing control to experiments to this distribution here and get a p-value. We're going to adjust the p-value using the Benjamin-Hochberg procedure to control the false t-square rate. So in the end, we can just threshold this list of p-value and get a list of experiments in which we think that cell motility is significantly different than the controls. So this leads us to the result. So on top of validating our workflow on simulated data, we have applied it to the MeterCheck data set, which was genome-wide RNA interference screen, whose goal was to find the genes that are involved in cell division. So it's constituted of 200,000 videos in which one gene of the human genome is downregulated. And as I was saying, so the application of Motif to this data set gives us a list of genes that we think are linked to cell motility, and we're currently working on this now. But something else that was permitted by the application of Motif to this workflow is to be able to answer the question whether there exists an ontology of cell trajectory in the data set. So namely, can we identify a certain number of groups of trajectories, cell trajectories in the data? And why was this permitted by our workflow? Well, we've been willing to answer this question right from the beginning, right? So you have cell trajectories, millions of them. Can you group them into groups of trajectories that are alike? I mean, why not? Well, why not? The problem is that we couldn't succeed when we pulled trajectories from all experiments because basically the data set was just too big, so it was just completely continuous. So the whole space of trajectories, so in the trajectory feeder space, was continuous, and we weren't able to identify any trajectory cluster. However, when we were pulling trajectories from heat experiments only, that is the experiments that were identified by the workflow as containing cell trajectories that are significantly different in terms of distributions from the control experiment, we were able to identify eight clusters. Oh no. Oh, well, sorry. So anyway, let's just look at the heat map. So that's just because I couldn't use my computer either. But anyway, so this heat map here gives you an idea of what the clusters are constituted of in terms of trajectories. So for each cluster, you have 1,000 trajectories that were picked at random. And the thing is that the color here indicates that we have here a cluster of very fast trajectories. And on the opposite side here, a cluster of very slow trajectories. But what's good is that in the middle, you have 6 trajectories that are completely unrelated to speed. As you can see here, what makes a difference is that, for example, here we have what we wanted to call Sturborn cells, as they have they're completely very more than average straight. And for example, another cluster here of shivering cells that have a high mean turning angle. But another question that we had following this study was to know whether the exposure of the cells to a small interfering RNA created new motile behaviors. That is, let's say in control videos, you have one main mode of cell motility, and siRNA exposure creates 7 other modes. Or do we simply have a redistributions in the cells in the different clusters following siRNA exposure? Well, this is what can be seen here. So for each cluster here, we have the distribution of trajectory percentage comparing control videos and experiments. And what we could see was that, in fact, when the cells are exposed to a small interfering RNA, it doesn't create new motility behavior, but rather the cells are redistributed in the different possibilities. So in conclusion, I've presented you with Motif, which is an integrated workflow for cell motility study in time-lapse data. Something that I haven't insisted on yet is that This workflow enabled us to obtain motility-related results from cell division screen reminding, which insists on the fact that it's also in the image bioimaging field very interesting to share and remind existing data. This workflow will be soon available in cell cognition, so stay tuned on the website. And right now, what we're currently doing is working on the set of genes that were identified by our workflow. In conclusion, I would like to thank the people I'm working with, especially Sandra and Thomas, and thank you for your attention.
Thank you very much. Do you have any questions? I had a question about the clustering. I didn't understand what you were saying about how initially it was one big ball and then you were able to pull out. Could you go over that again?
Yeah, so what I think happened is that when you pull trajectories from a whole data set, the biological variability makes that even though there might be clusters, the thing that is highly unbalanced and the thing that we have millions and millions of data points makes it that we feel the entire space that eventually exists between the cluster centers.
Oh, so you weren't visually able to see differences, but when you did the k-means, you got...
No, k-means didn't give any results, so we did cluster analysis. So basically we could compare the quality of the clustering in the two different cases. And actually you can have k-means work on millions and millions of data points because there exist like mini batch k-means that enable you to perform it. So it wasn't a technical problem, but really we couldn't, the quality of the clustering was really bad.
But then how did you end up with the 8?
So then when we were doing clustering on the subset of cell trajectories from the heat experiments, we could really get a nice curves that indicate that k equals 8.
Okay.
How well does your cell tracking cope with cell division?
Sorry?
How well does the algorithm cope with cell division?
Well, so basically, so that's the only prior information that you put. So you allow that cells can divide, merge for segmentation artifact, and appear, disappear, and do one-to-one. So that's hard-coded in the algorithm.
Thank you very much.